Hey everyone, let's study about Meselson and Stahl's experiment. Watson and Crick had hypothesized that the DNA replication was semi-conservative. However, there was no experimental evidence to test this hypothesis. So to test this hypothesis, Meselson and Stahl in the year 1958 designed a wonderful experiment. Now, the word semi-conservative means each of the strand is going to act as a template for the synthesis of new strand. And to prove if this phenomenon is true or not, we must have a method to distinguish between the parent strand and the daughter strand. Yes, dear student, you have heard correctly. To distinguish between parent strand and daughter strand. So here is what Meselson and Stahl did. They took the E. coli wild type cells and grew them in media containing N15 for several generations. And 15 is a non-radioactive heavy isotope of nitrogen. The presence of N15 will make all the molecules in cells, including the DNA, heavy. Now the question arises, what does it mean by heavy DNA? Students, it simply means if I take a E. coli cells and grown in N15 media and subject its DNA to density gradient centrifugation, then the density of this DNA will be more than the density of the DNA taken from cells grown on N14 media. The cells grown in N15 media were collected and transferred in fresh N14 media. So, as the cells divide, they incorporate N14, which is the normal isotope, into its DNA. The cells were harvested every 20 minutes and the DNA was isolated and subjected to density gradient centrifugation. Let's take a look at the results. The presence of only intermediate band after the first generation indicates that the heavy strand was acting as a template for the synthesis of new strands with N14 isotopes. As the cells divide further, more and more N14 is incorporated into its DNA. Just have a look over the picture. These results indicates that the replication of DNA is semi-conservative. That's all from my side. Thank you so much.